chapter 9. You remember that's about the cell cycle, cell reproduction, and our goal is to be able to describe mitosis and meiosis. Well, meiosis is chapter 10. So let's fast forward to where we left off. We were midway through talking about cancer, so that's where we'll pick up. Okay. Here's the good news about this, by the way, is that you get to fast forward me, you get to back me up, you get to make me repeat all you want. Write down questions that you have, because we will be, talking about this on Monday. And I'm very sorry there's been such a massive delay in getting this to you, but I appreciate your patience. And your next test will be open book. Okay, so where we left off, we talked about what causes cancer, and then we got sidetracked with the vaccines, that debate. So let's talk about cancer. Okay. Cancer is defined as the abnormal growth of cells, but just because it's abnormal does not make it a cancerous tumor. Now, generally speaking, an abnormal growth of cells is called a tumor, and you can start out with a benign tumor. It's not cancerous. It's encapsulated. That means that it has a distinct boundary. Benign tumors are very, very easy to cut out. They also don't invade neighboring tissues, and they don't spread. Benign tumors, they generally leave in place unless they cause you a problem, like maybe it's pushing on your spine or on your pituitary gland, which can cause gigantism. Okay, but for the most part, they'll leave a benign tumor in place. They will watch it like a hawk, though, because a benign tumor can develop into a malignant tumor. This is the one that's cancerous. Not encapsulated. It does not have a distinct edge. So they need to be careful when they cut them out to make sure that they got it all. Malignant tumors are also more than happy to invade neighboring tissues, and they also will detach and lodge into distant places. We call this metastasizing. Now, malignant tumors specifically result from mutation of genes that regulate the cell cycle. So cancer is by definition out of control cell division. Now, going from a benign tumor to a cancerous tumor, it tends to be gradual. It can take years or decades before a cell is obviously cancerous. So it's a good idea to stay on top of things. Now, here's a progression of cancer, and you'll notice up here at the top, there's this nice single sheet of cells, all nice and civilized, and one of them shows up with a mutation that causes it to start a tumor. So next picture down, we've got this nice little primary tumor. It's benign, it's not bothering anybody, but you'll notice the cells are piling up on each other. Now, out of nowhere, one of the cells will pick up a new mutation, and that's the purple cell there. And this particular cell is able to start invading neighboring tissues. I see a dolphin. This cell is able to invade neighboring tissues. Now, you'll notice on the third picture down, this one, that all the cells are purple. This is because with a tumor, the most aggressively dividing cell becomes the dominant cell in the tumor. You'll also notice it's more than happily invading all the neighboring tissues. And as a matter of fact, it's invading the lymphatic system and the blood vessels. Once the cells get into the lymphatic system or the blood system, the cells will frequently shred off and hitch a ride in either your blood system or your lymph system, one of the lymphatic ducts, and will then start a tumor elsewhere in the body. Okay, and that's what happens when a tumor metastasizes. It's not a new, different cancer. It's the same cancer. It's just set up someplace else in the body. Now, there are a couple of things that you see with all cancer cells. Okay, first off, they lack differentiation. They are completely non-specialized. If you look at tissue, normal healthy tissue, you'll see that liver cells look like liver cells and heart cells look like heart cells and lung cells look like lung cells. Cancer cells don't look like anything because they're not programmed to do anything. All they are is cancer. Okay. So this is lung cancer in situ, so in place. And you'll notice that the surrounding tissue is, well, kind of spongy looking. It's supposed to be. Its entire function is to fill up with air and get oxygen into your blood. Now that purple mass of cells right in the center is a cancer tumor. And you'll notice it doesn't look like any of the surrounding cells. Okay, that's cancer for you. Okay, also, cancer cells are immortal. Normal cells divide about 50 times and then they can't divide again. Cancer cells will divide forever. And to kind of illustrate that, let me introduce you to Henrietta Lacks. She had cervical cancer and she had surgery in 1951 to remove her tumor, and without her knowledge or consent, keep in mind this is the days before informed consent, her doctor took a sample of her tumor and threw it into a petri dish, and it grew. And this was huge because it was the first time human cells had ever grown in a petri dish ever. It became the first tissue culture line. They called it HeLa cells, H-E for Henrietta, L-A for Lacks. Now you'll notice her birth and death dates. She died in 1951. Her cervical cancer did kill her. So she was 31 years old, so she was young. Her tumor, on the other hand, she died, some 60 years ago. Her tumor is alive and well in just about every research lab on Earth. And to date, there have been 60,000 research articles printed, or submitted and printed, research using her cells. And every month another 300 are printed. To date, her research using her cells have led to 11,000 patents. Okay? She's long gone. Her tumor is alive and well. As far as we know, cancer has no expiration date. It's immortal. It'll just go forever. Okay. Now, along with cancer cells being non-specialized, they're really funny looking. They have abnormal nuclei. The nuclei may be enlarged. They might have a funky number of chromosomes. They also might have extra numbers of genes, even if they have the right number of chromosomes. They also do not undergo apoptosis. Okay? Normally, if there's damage to the DNA, you see apoptosis happening. Also, your immune system can recognize weird cells and can trigger apoptosis. It's part of their job, is to not only protect you from invaders, to protect you from damaged parts of yourself. Okay, cancer cells are abnormal, but they don't undergo apoptosis. They ignore any signals to undergo cell suicide. Okay, so that can be a problem because they'll just continue to divide. Okay. Cancer cells will also form tumors. Mitosis is normally controlled by contact inhibition, where a cell will divide until it runs into its neighbors, and then it'll stop dividing. It's already hogged up all the space that it's been allotted. Cancer cells, on the other hand, have lost contact inhibition. They'll also metastasize. The original tumor easily fragments, and you'll see new tumors of the same cancer appearing in other organs. That's how you wind up with like lung cancer in your brain or breast cancer in your leg. It's not a new cancer, same one. Also, here's an interesting one. Some cancer tumors are able to undergo something called angiogenesis because it's expensive to reproduce. It requires a lot of energy, it requires a lot of nutrients, a lot of oxygen. Now, cancer reproduces, it divides more aggressively than you do, which means its metabolic needs are higher than yours. Okay, so if certain tumors are able to spark a mutation where it's able to induce your circulatory system to build blood vessels towards the tumor, and that brings nutrients and oxygen to the tumor. And sometimes when you see someone wasting away when they have cancer, sometimes it's from the chemotherapy. It does wreck your appetite. Sometimes it's the cancer itself, because all the food and oxygen they're bringing in, the cancer is taking it. Okay, some cancer treatments actually aim towards preventing angiogenesis, starve the tumor, cause it to shrink. Okay. Here's a beautiful table out of your textbook that kind of brings on to one slide what I was just talking about. So you can pause and look at this. Okay, so as far as what exactly gets mutated to cause this, well, mutations happen all the time. But most of the time, a mutation doesn't cause cancer. There are two classes of genes that are involved in cancer. There are proto-oncogenes. These are genes that encode proteins that make the cell cycle go. I think of them as the gas pedal. These are the proteins that tell the cell cycle to go, that tell it to speed up. When these mutate, they become oncogenes. These are cancerous genes. When they are oncogenes, they are cancerous. And when they mutate, they get stuck in a position where they're constantly on. So all the cell cycle is hearing is go, 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 faster, faster, faster. The other type of gene that gets mutated is tumor suppressor genes. And these normally turn cell division off or slow it down in healthy cells. And P53 is a tumor suppressor gene. When these mutate, they get stuck off, so they can't do their job. So I think of these as normally the brake pedal. They're like stop, slow down. And yeah, when these break, it gets ugly. Now, a really nasty, aggressive cancer will have mutations in both the proto-oncogenes and the tumor suppressor genes. So all the body's hearing is, divide, divide, more, more, and it's not hearing any slow down, ease off, stop. Also, another source of, well, this one actually accounts for the immortality of cancer, is chromosomes have special material at the end of the chromosomes called telomeres. And what they are is extra DNA at the end of the chromosomes that is repeated. And it doesn't even matter what the sequence is. It's basically junk DNA. Its function is structural. Okay, every time the cell divides, the telomeres get shorter due to the polymerase. That's the enzyme that actually does the copying. The polymerase can only copy in front of it. It can't copy the DNA that it's sitting on. So every time the cell doubles, every time the DNA copies itself, you lose about 100 bases. When you run out of these telomeres, their entire function is to get cut off a little bit at a time. And when you run out of them, the cell line stops dividing. Okay. So when these telomeres get very short, the cell won't divide anymore. Now, in stem cells, you have these cells that will divide throughout the rest of your life. They handle repair of injury. Anyway, they have an enzyme in them called telomerase that repairs the ends of the chromosomes every time the cells divide. Now, when the telomerase gene mutates, that causes the telomerase genes to be turned on where they shouldn't be, and that causes the telomeres to continue to lengthen. Every time the cancer cell divides, the telomeres get repaired, and that allows the cancer cells to continually divide. Okay.
The ends of chromosomes contain regions referred to as telomeres. These telomeric regions consist of telomeric repeat sequences. The exact sequence of the telomeric repeat can vary from species to species. The repeat sequence shown here is found in the telomeres of human chromosomes. Since DNA replication can only proceed in the 5' to 3' direction and requires a primer, it is not possible for a DNA polymerase to replicate the 3' ends of linear chromosomes. If this problem were not corrected, chromosomes would shorten with each cycle of DNA replication. The addition of telomeric repeat sequences by an enzyme complex referred to as telomerase assures that this shortening does not occur. Telomerase is a unique and interesting enzyme because in addition to having DNA polymerase activity, it also contains an RNA sequence that provides a template for the synthesis of telomeric repeat DNA. Part of the RNA sequence hybridizes with the single-stranded overhang on the DNA strand, leaving a single-stranded overhanging RNA sequence. The repeat unit shown here is from Tetrahymena, which is a model organism used in telomere and telomerase research. The DNA polymerase function of telomerase then synthesizes the DNA strand complementary to the RNA found in telomerase and subsequently translocates to the end of the newly synthesized strand, and the process repeats itself. Many cycles of repeats can occur. Once telomerase has completed its function, DNA primase synthesizes an RNA primer near the 3' end, and DNA polymerase fills in the vacant region. A short region at the 3' end will remain single-stranded. The end result is that telomerase will have added many repeat sequences, from a few dozen to several hundred. This prevents chromosome shortening.
So what we do with mitosis as adults, it enables growth and repair. In flowering plants, they have stem cells too, but they get a special word for it, meristem. You see meristem around the girth of a big tree. That's how tree trunks get thicker, and also at the ends of sticks. That's how the plant grows new twigs, grows flowers, grows leaves, all that. Now, as far as us, animals, we use mitosis when a fertilized egg, that would be a zygote, becomes an embryo. The embryo then develops into a fetus. The fetus becomes a baby who becomes an adult. Also, every time a cut heals, a broken bone mends, a burn heals, anytime you have healing or repair of damage, that's all mitosis. Okay. Again, lovely table out of your textbook, and it's a all-in-one view of general cell division, who uses what version and what they use it for. Notice that prokaryotes and eukaryotes both use standard cell division for asexual reproduction, or just splitting into two individuals, but they do it via different methodologies. Eukaryotes do mitosis and cytokinesis. Multicellular ones use it for development, growth, and repair, but single-celled, that's how they make more of them. All right, so yay.